This lizard only eats 10 times a year. And that single fact created a drug worth $50 billion. This is one of the most incredible discovery stories in modern medicine, and it almost never happened. Ozempic wasn't invented, it was discovered. Your body already makes a version of Ozempic. It's called GLP-1, a hormone your gut releases after you eat. It tells your pancreas to make insulin. It signals your brain that you're full. It does exactly what Ozempic does. So why can't we just use that? Here's the problem. GLP-1 lasts about 2 minutes in your bloodstream. 2 minutes. Then it's gone. Broken down, useless. You can't build a drug around something that disappears in 2 minutes. By the time it reaches your bloodstream, it's already gone. Scientists knew this for decades. They knew that if they could find a version of GLP-1 that lasted longer, hours instead of minutes, they could treat diabetes, maybe even obesity. But human biology wasn't cooperating. The answer wasn't going to come from a lab, it was going to come from here, the Sonoran Desert, where something had already solved this problem. Something ancient, something venomous, something that had been perfecting this chemistry for 20 million years. It's 1980. A young gastroenterologist named Jean-Pierre Ralphman has just arrived at the National Institutes of Health in Bethesda, Maryland, and he's been given an unusual assignment. Test different animal venoms on guinea pig pancreases. See what happens. This wasn't targeted drug development. This was, let's squirt poison on organs and see what reacts, science. But sometimes that's how discoveries work. Ralphman ordered powdered venoms from a place called the Miami Serpentarium and started testing. Bee venom, nothing special. Snake venom, interesting but inconsistent. And then, Gila monster venom. The pancreas went crazy. Insulin production spiked. The cells lit up like a Christmas tree. But why? Why would a venomous lizard from the Arizona desert have a molecule that perfectly stimulates a mammalian pancreas? The answer is one of the most elegant pieces of evolutionary engineering you'll ever hear. Meet the Gila monster, one of only 2 venomous lizards in North America. Slow, lumbering, covered in beaded scales that look like they were designed by a jeweler with a dark sense of humor. And here's the thing about Gila monsters. They're lazy. Not in a bad way, in a brilliant way. A Gila monster might eat only 3 to 10 times per year. Let that sink in. They gorge themselves on bird eggs, baby rabbits, whatever they can find, sometimes consuming 1/3 of their body weight in a single meal. And then they wait for months. Now here's the problem this creates. When you go months without eating, your blood sugar should crash. Your body should panic. Things should go very wrong very quickly. But the Gila monster? Perfectly stable. Months of fasting and its blood sugar barely budges. How? Evolution built a solution directly into its saliva, a hormone called Exendin-4, 53% identical to human GLP-1. But with one crucial difference. Human GLP-1 lasts 2 minutes. Exendin-4 lasts hours. The Gila monster evolved the exact molecule scientists had been searching for because it needed it to survive. A venomous desert lizard had solved a problem that the best human scientists couldn't. But finding the molecule was only half the story. Now someone had to fight for it. Word of Ralphman's strange pancreas experiments eventually reached a researcher named John Eng. Eng was an endocrinologist at the VA Medical Center in the Bronx, and he became obsessed. He ordered his own Gila monster venom from the Miami Serpentarium, isolated the active compound, characterized its structure, and in the early 1990s, he made it official. Exendin-4. A hormone from lizard spit that could do what human biology couldn't. The science was solid. The molecule worked. The discovery was real. Now came the hard part, convincing anyone to believe him. The concern? A molecule from a foreign species might trigger a destructive immune response. It was too risky, too weird. Nobody wanted to stake their company's reputation on a lizard. Eng's own medical center wouldn't even help him file the patent. By the mid-1990s, he was desperate. Colleagues remember him being close to tears at scientific meetings in Europe. Picture it, a fluorescent-lit conference hall, rows of research posters, scientists walking past without stopping, and John Eng standing next to his poster on Exenatide 4, waiting for someone, anyone, to care. The man who had discovered what would become one of the most valuable drug classes in history couldn't give it away. Then, in September 1996, at the American Diabetes Association annual meeting in San Francisco, a man named Andrew Young walked by Eng's poster. Young was the head of physiology at a small biotech company called Amylin Pharmaceuticals. He read the poster, he saw the data, and unlike everyone else, he believed. Amylin took the chance no one else would. They licensed Exenatide 4, ran the trials, proved it was safe in humans. And in 2005, the FDA approved Exenatide, brand name Byetta, as the first drug derived from Gila monster research. But that was just the beginning. Because once you prove the concept works, others follow. Novo Nordisk took the idea further. They engineered semaglutide, a modified version of human GLP-1 that lasts even longer. In 2017, it launched as Ozempic. Now, you're not literally injecting lizard venom. Ozempic is synthetic, built in a lab. But it only exists because a Gila monster proved it was possible. The molecule no one wanted became a $50 billion drug. The poisonous lizard that pharmaceutical companies laughed at had spawned a drug class that would change medicine forever. Today, GLP-1 drugs are being tested for things no one imagined: addiction, Alzheimer's, heart disease, Parkinson's. So the next time someone tells you an idea is too weird, too unconventional, too crazy to work, remember John Eng, standing alone at a conference, holding research no one wanted on a molecule from a lizard.